Hello and welcome back to Biochemistry for Health Sciences channel. In the previous videos, we talked about electrolytes, and then we talked about calcium and phosphate. So these are all the inorganic chemicals, some of the medically relevant inorganic chemicals found in our body. And remember, these videos are just introductory videos that give you a basic background on the chemistry that will become useful to understand the physiology as well as the pathophysiology involved with these chemicals in future videos. So in this video, we're going to talk a little bit about the some basic chemistry of iron so that we can appreciate some of its properties in biomolecules as well as its clinical significance. So if you look at the way electrons are arranged in, say, calcium, you can see 1s2, 2s2, 2p6, the different orbitals, 3s2, 3p6, 4s2, and so on. Now, iron has an atomic number 26, and we arrange the electrons in this iron, following the rules of chemistry. We now get into the d orbitals. D orbitals. See, and there are 5 of them. Okay, so the iron atom has this electron configuration. So usually we write the D6 first and then the 4s2. A little switch because of the way the energy of these orbitals are. So that's an iron atom. Now of Fe2 plus loses 2 electrons, basically loses these 2 electrons. So the electron configuration is this. These 2 electrons are the outermost electrons that are lost. Fe3 plus, the ferric, this is the ferrous, this is the ferric. It has lost 3 electrons, so 26 minus 3 is 23 electrons. And when you arrange the 23 electrons, this is the electron configuration that you get. Now, there is a reason why I wrote the electron address or electron configuration for the noble gas krypton, okay, which has 36 electrons. We'll talk about that in a few minutes. 36 electrons. So one of the most important functions of iron is in oxygen transport, and as we mentioned in the earlier video, oxygen and transport is all about energy, and continuous energy production is very, very, very important for our body, for our cells. So the protein hemoglobin, which carries oxygen in the red blood cells, as well as the protein myoglobin, which is found in muscles, which also binds oxygen. These proteins have the iron atom, and this iron atom is attached to what we call the heme group. Okay, this is an organic molecule that is red in color. And that's one of the major reasons why your blood is red. It's because of this thing called heme, this organic molecule. So the iron is attached to the heme group. Okay, so this iron is in the form of Fe2 plus, the ferrous. So the ferrous is what you find in the heme of hemoglobin and myoglobin. And the heme group, together with the iron, is located at what we call the active site. We'll talk about this under proteins. The active site of the protein, whether it's myoglobin or whether it's hemoglobin, the heme group is at the active site of the protein. So the protein gives the whole heme iron a very nice environment. A very nice environment so that the iron can do its job. And what is its job? Its job is to bind oxygen. Okay, bind oxygen. So the ferrous, you can see the ferrous, the Fe2 plus, which is what we see in our heme group in myoglobin and hemoglobin. The ferrous ion, you can see, has 24 electrons. So all these elements, they try to become noble. Okay, so the nearest noble element for ferrous is krypton, which has 36 electrons. So 36 minus 24, that's 12 electrons. So this ferrous is going to try to get 12 more electrons when it interacts with other chemicals. So in this case, you can see the ferrous interacting with the heme, 2 electrons, 4 electrons, each bond is 2 electrons, 6 electrons, 8 electrons. And then it also interacts with a part of a protein, a histidine of a protein. So that's a total of 10 electrons. And then finally it can interact with in this case, oxygen, and that will give it the 12 electrons to reach the noble, to be part of a royal family, the noble krypton. So now it's very happy. So that's why this ferrous can form, you can see 1, 2, 3, 4, 5, 6. It can form 6 bonds basically with different atoms. So this ion chemistry is very complex. It's fascinating, but it's very complex, and chemists are still trying to totally understand. But one thing we do see is, for example, in hemoglobin that doesn't have oxygen bound, what we call deoxyhemoglobin, that don't have oxygen bound. You can see that the color of deoxyhemoglobin is sort of dullish red. And that's because the shape of the heme is different. Because remember, the iron is interacting with the heme. Okay, the iron is interacting with the heme. Okay, so that's because the iron atom, when it's not bound, the iron atom, when it's not bound, is larger. That's because the way these iron atoms, the iron electrons are arranged in the d orbitals. You can see the d orbitals are slightly different. Okay, we call that the high spin and the low spin. So you can see these 5 d orbitals have slightly different energies. These 2 are different, these 3 are different. Okay, so because there are more lone pair electrons, lone pair, all alone in their room, so there's more repulsion, there's more lone pairs, so the iron atom is a bit larger. So this iron atom here is a bit larger in deoxyhemoglobin. And that causes the heme to be slightly of a different shape. It's more of dome shape. And when the shape of an organic chemical changes, the properties will change too. Okay, so the color in this case is a property. So the heme is more dullish red, sort of dullish red in color. However, when oxygen is bound, when there is oxygen bound to the iron atom in heme, whether it's myoglobin or hemoglobin, in this case hemoglobin, okay, remember the iron is still Fe2 plus. When oxygen is bound, then we get oxyhemoglobin. And now what happens is the iron atom, the way the electrons are arranged changes. Okay, so you can see there are changes. Here, now these 3 pairs, there are no more lone pairs. Okay, so there's less repulsion, there's no lone pairs. So the iron atom radius changes, it becomes smaller. And since the iron is attached to the heme now becomes flat. And since heme is colored red, the color of the heme changes from dull red to bright red. So that's why oxyhemoglobin, oxygenated hemoglobin, it has a brighter red color compared to deoxyhemoglobin. Okay, so a very fancy chemistry that we talk about high spin and low spin and so on. But in terms of clinical significance or biological significance, one of the things we see is the color of hemoglobin. Deoxy is dull red, oxyhemoglobin is bright red. Heme is very important for oxygen carrying capacity of the body, and so is iron. Now, I would like to mention a little bit about this type of hemoglobin where the iron is in the oxidized ferric form. So there's 23 electrons. So basically there are only 5 electrons in the d orbitals. So you can see these 5 electrons in the d orbitals. This is called methemoglobin. Okay, it's not very good in carrying oxygen. It's because the iron has been oxidized. Okay, so normally we should have less than 1 to 2% of a hemoglobin where the iron is in the ferric form. Okay, but there are certain cases, there are some genetic diseases or congenital reasons where this amount can rise as high as 20%. So 20% of the hemoglobin in these patients may be in the methemoglobin form. And in that case, of course, there'll be more deoxyhemoglobin, and so there's a sort of mild cyanosis. The symptoms are mild to severe, and of course, because hemoglobin is important for energy transfer, these symptoms appear because of the lack of energy. There are some drugs that can, including nitrates and nitrites, that can cause your hemoglobin to become methemoglobin. Basically, the Fe2 plus gets oxidized to Fe3 plus. And in case of acute toxicity, the levels of methemoglobin can go up as much as 40-50%. And that can be very dangerous. Okay, that can be fatal. Okay, so methemoglobin is another test that is sometimes done in certain cases. So the iron in our body, okay, we have a certain amount of iron in our body. Males have more, females have slightly less because of menstruation. So the iron can be basically divided into 2 types, storage iron and functional iron. So 80% is functional, 20% is storage. Ferritin is the soluble storage iron. Hemosiderin is the insoluble storage iron. And storage iron is mostly found in the liver, over 60%, and in other cells such as your macrophages. Functional iron, as we mentioned, hemoglobin is about 80% of the functional iron. 80% of this 80%. Myoglobin has about 15% of the functional iron. And then there are many other proteins, cytochromes, which we'll talk about under the electron transport chain or the respiratory chain. Then the iron sulfur proteins, which are also part of the respiratory chain, they all have iron in them. Okay, and then enzymes such as peroxidase, and then there's this enzymes called cytochrome P450s, which are very rich in the liver that help detoxify drugs and all kinds of chemicals. So that's functional iron. Important to know that in your blood, in your serum, the iron, the ferric iron, is bound to a protein called transferrin. Okay, that's how we transport iron from in the blood from one tissue to another. Transferrin. Okay, so these words here, ferritin and transferrin, are colored in blue because these are very important lab tests, which we'll talk about in a few seconds. I should let you know too, a warning, free iron, especially the Fe2 plus, is dangerous. It can form free radicals. And that's why we make sure that all our iron is bound to a protein, in this case, transferrin. Okay, so that's why iron has to be bound. Free iron can actually cause problems because it generates free radicals. Okay, so iron, you can see, is necessary for proteins like hemoglobin and myoglobin to bind oxygen and transport oxygen because of the d orbitals are able to help the iron to bind many different atoms at the same time. So in this case, binding the protein, binding the heme, and then binding the oxygen. Besides transport, there's another very important function of iron, and that is the fact that these transition elements such as iron can exist as stable atoms or ions, whether they are reduced or whether they are oxidized. And this is very important when we talk about the electron transport chain. This is very important in redox reactions, and especially in energy generation. Okay, the respiratory chain is very important in energy generation. So for example, Fe2+, as we saw, has 24 electrons. Okay, now it can donate its electron in the electron transport chain and eventually to oxygen. Okay, so oxygen is the final acceptor of electrons. And when it donates the electron, it becomes Fe3+, ferric. It gets oxidized. But then if this ferric is given an electron, you see 3+, you give one negative electron, it becomes 2+. It goes back. Okay, so it can give electrons and become this. It can take electrons and become this. So that's the beauty about ferric. It can give electrons, take electrons, give electrons, take electrons. And this cycle allows the ferric to work as a very nice atom for transferring electrons from one chemical to another. And these are called redox reactions, right? Redox reactions are simply reactions where electrons are transferred from a certain atom in a chemical to another atom in a chemical. Redox reactions. Okay, what about the lab tests for iron? There are several lab tests for iron. So let's look at some of this. One is ferritin, serum ferritin. You can see it's the levels are very low, micrograms per liter. Ferritin is the most sensitive indicator for iron deficiency anemia. Okay, so ferritin is what decreases the first whenever there is iron deficiency. And iron deficiency, unfortunately, is a very common nutritional problem in the world. Okay, so ferritin is one test that we do in the hospital lab to help physicians diagnose the reason for anemia. And we'll talk about anemia and all the chemistry that can cause anemia much later in our future videos. Then, of course, we can measure serum iron. We can measure the iron that is bound to the transferrin. Then we measure transferrin. Okay, the amount of the protein transferrin in the serum, we can actually measure the protein. And then we have a test called TIBC, total iron binding capacity. Okay, one transferrin binds 2 iron atoms. Okay, so that's why this value is about twice. You can see twice the value of the transferrin. And then we have, when we'll talk about this, we'll explain these 3 tests here at the bottom so you can understand what these tests mean. Then we have another test called percent transferrin saturation. Basically, take the serum iron, divided by the TIBC. Okay, so you take the serum iron divided by TIBC, like 11/43 times 100, and you get a percentage. The reference range is around 20 to 50. And then we have what we call the unsaturated iron binding capacity, or UIBC. Okay, UIBC. And basically, the TIBC is equal to serum iron plus UIBC. Okay, so to understand this, let's use a simple cartoon here. So here we have, let's say, just for simplicity, there is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Let's say there are 10 transferrin molecules. And each transferrin has 2 binding sites for iron. So the total iron binding capacity is 20. Of course, these are just numbers we use here, but in reality, you use concentrations, right? You use concentrations in terms of either milligrams per deciliter, or micromolar, or millimolar, or whatever. Okay, so if you had 10 molecules of transferrin in the sample, the TIBC would be 20. Let's say the iron amount was 6. 1, 2, 3, 4, 5, 6. Okay, so the percent saturation is how many ions, concentration of iron, 6, divided by TIBC, 20 times 100. So it comes out to be 33%. That means 33% of all the possible binding sites for transferrin are bound by iron. Okay, that's the bound form, 33%. Percent saturation, percent bound saturation. So UIBC, remember there are 20 sites, okay, 6 are bound. So UIBC, there are 6 bound, there's 20 TIBC. So UIBC is just 20 minus 6, and that comes out to be 14. And that's called the unsaturated iron binding capacity. That means how much more iron can be bound in case your iron levels go up. Do you have enough transferrin to bind that iron? Because we don't want free iron. Remember, we do not want free iron. Especially in the Fe2 plus form in inflammation, we realize that one of the problems we might have in chronic inflammation is the free iron, Fe2 plus, is actually forming free radicals. Very dangerous free radicals. Okay, so this is a no-no for the body. So that's why only in the normal case you can see only about 20 to 50% of your transferrin is saturated. So we have a lot of extra space. You can see extra space to bind our iron in case we have too much iron. Okay, in case we have too much iron. And how do you know that iron is bad? Well, there are certain diseases. One is called hemochromatosis, we'll talk about later. The patient has too much iron, and these patients around the time they are maybe 25, 30, 35, very early in life, they get their probability of getting a heart attack is very, very high. Okay, so that's why we know that free iron can be very bad for our tissues as well as our blood. Okay, so that's all for now. We hope you enjoyed this video. Again, these are just introductory videos. We'll talk more about the ADME of iron and all these other inorganic chemicals in the past videos, as well as the pathophysiology. You know, why would someone get high iron or low iron concentration and so on in the future videos. Okay, until next time, goodbye.